Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 15th July 2020. From our memory we understand that we were discussing and reflecting on cell and its organelles, and in our previous session we discussed about the structure of plasma membrane. And remember, Nicholson, Singer and Nicholson model, fluid mosaic model. That's very important one. I hope you have drawn diagram and labeled it and understand each label of it. And in this session today, we are going to talk about another important organelles called plastids. And basically, whenever we talk of plastids, we remember food. They are food storage organelles, and they are largest cell organelle present in plant cell. So they are not found in animal cells. Plastids basically are of three types. I'll start off with the one that you are very familiar, chloroplast. Green plastids, their color is green, chloroplast. Having green color, they help in photosynthesis. And why do they have green color? Green color of chloroplast is because of the presence of a pigment called chlorophyll. And chlorophyll is characteristically green in color. So green colored pigment called chlorophyll is present in chloroplast, and that is the reason why we call them as green plastids. And they are present in mesophyll cells of the leaves, green stems containing chlorophyll, and of course rest of the plant except root, these will be present maximum. And you know that chlorophyll absorbs sunlight, and you have studied in class 10th light reaction and dark reaction, and you know that chloroplast is very essential for photosynthesis. And next familiar one is chromoplast. Chromoplast means colored plastids. You are seeing every day in your garden, in the environment, in and around your home, that there are plants having red colored flowers, orange colored flowers, yellow, violet. Similarly, in some plants like crotons, you are seeing red colored leaves, orange colored leaves, yellow colored leaves, violet. So all these colors other than green are because of the chromoplasts. And there is something called keratin, xanthophyll. So here we said chlorophyll, green color. Here xanthophyll, yellow. Anthocyanin. These are the various pigments, coloring substances that are present in chromoplast. That's why chromoplasts are responsible for various colors present in flower as well as leaf, and in some extent to stems also. And they also store starch and help the plant to utilize the stored starch. Similarly, leucoplast. And leucoplasts are colorless plastids. Remember, chloroplast green color, chromoplast varieties of colors, and leucoplast no color at all. But they perform an important function called storage of starch, and they are present in all the modified underground versions such as tubers, roots, etc. So basically plastids are of three types: chloroplast, chromoplast and leucoplast. And what about structure? Chloroplast is enveloped by double membranous sac. Two layers will be there. It encloses a liquid which has got lot of proteins, and that liquid in general we call it matrix. Common name is matrix, but in particular in chloroplast it is called stroma. So stroma is the liquid portion having lot of proteins within the chloroplast, and within this matrix there will be a lot of finger-like structures. They are called tube-like structures or tubelets, lamellae. It is denser in certain places due to pigments grana stacked in group, and you can see virtually the figure. And this is how the original one, green color chloroplast, and you can see the stroma. This is the liquid present within this, and double membranous outer membrane and inner membrane. And this within the stroma, that liquid portion of the chloroplast is called stroma. Stroma has got a lot of proteins, and in this liquid, proteinaceous liquid, you can see disc shaped structures. You are seeing here, just like you have to imagine. Take 1 rupee coins and place many 1 rupee coins in your palm. You get a row or stock of 1 rupee coins. So the granum, singular, grana, plural, granum will be like that. And each 1 rupee coin which you are holding is called thylakoid. So many thylakoids will be placed one above the other, that becomes granum, and many granum will be there within the cytoplasm or stroma of the chloroplast, and many grana will be floating within the stroma, and each granum will be interconnected. See here, interconnections here, granum to granum, granum to granum, granum to granum. These are called lamellae, granal lamellae. You can also see here same structure, outer layer, inner membrane. Then you can see thylakoids, you can see grana, you can see the stroma, and then you can also see many proteinaceous structures within the stroma. So the most important characteristic that you must remember is that this organelle within the stroma has nuclear material. That is important. Normally cell itself will be having DNA and RNA, but two cell organelles which are very, very important having DNA and RNA are this chloroplast, and then next one we will discuss mitochondria. Mitochondria and chloroplast will have their own DNA and RNA. So many in UPSC exams, CBSE exam, this question will be asked. So keep remembering chloroplast and mitochondria. And then ribosomes, fat globules, starch, a number of enzymes. So the main function of chloroplast is photosynthesis. Remember in your 10th class you studied light reaction, dark reaction, and light center, dark center, 740 nanometers. You said PS1, PS2, 780 nanometers. And this 740, 780 nanometers of wavelengths of sunlight is absorbed by chlorophyll, and the chlorophyll is present in chloroplast. That is the reason why we say that chloroplasts help in photosynthesis. And next we move on to another important organelle within the cell that is present is mitochondria. And chloroplasts or plastids are helping in synthesis and storage of food, which is starch, which is the food of the plant. We say that they are called kitchens of the cell. Kitchens of the cell or storehouse, food godowns of the cell. And the mitochondria, once that food gets broken, glucose is formed, and that glucose will be again broken, and one glucose molecule will give an organism or the cell 36 ATP molecules of energy. ATP, adenosine triphosphate, and that is chemical form of energy. So we say mitochondria are the powerhouses of the cell, just like Karnataka Electricity Board. So they are powerhouses of the cell. Why they are called powerhouses of the cell? Because they produce ATP, energy. And we say ATP is the energy currency, like Indian currency is rupee. Like that, cell currency is ATP. So we say energy currency is ATP. And mitochondria, of course, this much long, and they are spherical or rod shaped, and they are surrounded by two membranes, that is outer membrane and inner membrane. Inner membrane, the characteristic of this mitochondria is the inner membrane gets inward, deep into matrix. Matrix is the liquid that is present inside the mitochondria, and extend themselves and form what is called cristae, that finger-like structures. The inner membranous space is filled with a liquid, and inner membrane has large surface area than outer membrane. So particles present on the inner membrane are called oxysomes, and very commonly, famously, they are called F1 particles. F1 particles. These particles play important role in what is called electron transport, which we will be studying in plant physiology. In detail, we will be dealing with this electron transport, how the electrons will pass on from one source to another source, and during this passage of electrons or transportation of electrons, how energy will be released, what is the fate of ATP, and how the finally cell gets energy. All that we will discuss in electron transport. And so matrix of mitochondria has a lot of enzymes, and these enzymes help in again respiratory cycle called Krebs cycle or TCA cycle, ATP synthesis, as I said just now, and protein and fat synthesis containing DNA, RNA and ribosomes. Now, having so much of importance, let us see the structure. When you take the vertical section of the mitochondria, this is how it looks like. Now we will go from outer to inner, and outside you are seeing a membrane, outer membrane, it's a spherical or ovoid, and inner membrane. And what I want you to see is this inner membrane is getting inward foldings. Look at that, this inward foldings, and these inward foldings which are entering into the matrix, the inner liquid. Each inward folding of the inner membrane is cristae. This is a cristae, this is a cristae, this is a cristae, this is cristae, so as this. And within this cristae, finger-like structure, look at that, there is F1 particle, that oxysomes we say. So inside you got to matrix, and this matrix will be having many enzymes. So these enzymes play very crucial role in very important physiological functions such as Krebs cycle. So mitochondria contains 70%, look at that, 70% of the enzymes of the cell, and is mainly connected with a very important function called respiration. And because of respiration, just now I said, the glycolysis takes place, glucose will be broken down, and whenever the glucose is getting broken down, we get a lot of energy. Per glucose molecule, I said 36 ATPs will be released, and ATP is adenosine triphosphate. And mitochondria totally, look at that, 70 enzymes and 14 coenzymes. That is the importance. So what is that you have to do? Draw the diagram, label it, and after labeling, you just talk about what is the importance of outer membrane, inner membrane, what are cristae, what is matrix, what is F1 particle, and so on and so forth. And we must be thankful to this lady, American biologist Lynn Magles, provided evidence that some organelles within cells were at one time free-living cells themselves. They are free-living because there is a evidence, scientific evidence, that these have got their own DNA. That is the reason why I said in many exams this will be asked. Organelles that have DNA. So one is chloroplast, another is mitochondria. Remember this. And both chloroplast and mitochondria are semi-autonomous, as they have DNA, genetic material, RNA, and ribosomes, protein synthesizing organelles. That is the reason why mitochondria and chloroplast are very important. And now we go to third one, that's endoplasmic reticulum. It's called ER. And this ER, thanks to this gentleman, Porter and others, in 1945, reported and described structure of endoplasmic reticulum for the first time. The plasma membrane is folded are extended inwards into the cytoplasm in the form of double membranous system of thousands of tiny tubules. They may be branched or unbranched. This membranous system extends from plasma membrane up to nuclear membrane. Like this is the cell and this is the nucleus, so extension. The plasma membrane is getting extended towards the nuclear membrane. That means we can say endoplasmic reticulum is a bridge, just like a railway bridge, connecting plasma membrane with nuclear membrane. So this is the plasma membrane and connecting to the nuclear membrane. That's why endoplasmic reticulum is this. And it is of two types. That is smooth endoplasmic reticulum, rough endoplasmic reticulum. So you got basically endoplasmic reticulum. This is smooth endoplasmic reticulum. And you got on the endoplasmic reticulum ribosomes, then we say it is rough endoplasmic reticulum. So on rough endoplasmic, there is always presence of ribosomes and microsomes. Well, let's see the diagram. And the space enclosed by tubules is cisternae. Now look at this diagram. This is the nuclear membrane, and this is the plasma cell membrane. Now you can see the extensions. You can see here clearly. This is the nuclear membrane, and the nuclear membrane is getting extensions and forming tubules or tubulets. Here also you can see these are called cisternae. And the cisternae, of course, we will be studying that in future they get cut off and form round structures, vesicles. So you got cisternae, tube-like structures, and vesicles, as you are seeing here. These are all vesicles, round structures. So these will become in future vacuoles, and the vacuoles can be food vacuoles or excretory vacuoles. So endoplasmic reticulum is playing a very crucial role in this in two ways. One, it is providing vesicles, and second, it is giving place for ribosomes to lay down there. So ribosomes play a very crucial role. They form proteins, hence protein synthesis takes place because of ribosomes, and ribosomes are because of endoplasmic reticulum. So you can see here, and these ribosomes are very, very important. Depending upon their size, there are prokaryotic 70S ribosome, and so this complete thing is a 70S, and this 70S is divided to two areas, a 50S part and a 30S part, and depending upon, as I said, how many proteins it can synthesize. Eukaryotic, like in our cells, there is a 80S ribosome, and that 80S, one part, bigger part is 60S and smaller part is 40S. But please remember, this is not mathematics calculation. Don't say 60 plus 40, 100, how it can be 80. No, these are units, that's all, okay? And this 60S again, you can see to the tune of 45 proteins, and 40S to the tune of 30 proteins. So functions of endoplasmic, as I said, mechanical support and act as cytoskeleton of the cell, divides the cytoplasm into number of compartments, and a lot of enzymatic actions will take place. Cisternae will be formed, and they help in transport of metabolites and the cytoplasm to nucleus and in between cells through pits, help in synthesis of sterols and phospholipids, excretion of metabolites outside plasma membrane, and holds ribosomes for synthesis of secretory proteins. So we'll talk about how exactly ribosomes will help in protein synthesis